Hello, my name is Michael Corselli. I'm the founder and chairman of Flya. I want to welcome everyone to today's webcast, which will be questions for me and answers from Dr. Reid Rubsamen, CEO and founder of Flow Pharma and Strategic Vaccines, with Henry Schoenberger. He's a senior advisor to Strategic Vaccines CEO, asking Reid to elaborate when necessary. Reid Rubsamen graduated from Berkeley with an undergraduate degree in biochemistry and computer science. He received his MD from Stanford and an MS in computer science. After medical school, he did his residency at Harvard Mass General in anesthesiology, which he was the chief resident. At MIT, he participated in an NIH-funded fellowship in computer science. Then he received a master's degree from Harvard in healthcare management. Today, he has 71 US issued patents. His work has been cited over 10,000 times in R&D researchers' articles. 20 years ago, he pioneered an inhalation therapeutic for diabetes. And 10 years ago, with this reasoning from what he knew about SARS-1, developed a revolutionary targeted T-cell platform to combat a quickly spreading lethal coronavirus that he postulated would cause a future pandemic. At the same time, Reid continues to practice anesthesiology part-time and teaches residents at university hospitals, Cleveland Medical Center. Reid's primary motivation is his dedication to saving lives. His past is a prologue to his overwhelming commitment to defeating critical disease. Today, Reid will explain why we need a revolutionary 21st century immunology platform to stop a 21st century deadly virus engineered to evade defeat, and why the 20th century antibody platforms that have historically been efficacious are not sufficient to stop the constantly shape-changing COVID-19 variants. Further, Reid will explain why a targeted killer T-cell platform will defeat the Delta and future variants. A little bit about Henry Schoenberger's background. It's not in bioscience. However, since October of 2020, he has learned enough from working directly with Reid and his team to coordinate and develop information to provide communications, which adds clarity to the crucial need for the revolutionary targeted killer T-cell platform Reid created 10 years ago. Dr. Rubsamen's scientific breakthrough has resulted in the only developed killer T-cell therapeutic that will immediately arrest the COVID Delta variant and future variants in people just testing positive, and provide long-term immunity because it is actually a vaccine. Henry can ask Reid follow-up questions as we go through our fireside chat today, which he'll know will help elicit a burst of insight from Reid. But before we get into the Q&A, I want to play a 1-minute video from our host today, Strategic Vaccines. Kevin, will you play the video?
I'm Reid Rubsamen. I'm the CEO of Flow Pharma and Strategic Vaccines. We have developed the FlowVid20 vaccine for SARS-CoV-2. What we're doing is different. We're the leaders in the space. We are the only group in the world with an advanced stage developed T-cell vaccine for COVID, for SARS-2, the virus that causes COVID-19, with data in the gold standard rhesus macaque model showing that this is protective against very high doses of SARS-2. We're able to give this drug as a dry powder that is room temperature stable. This gives us a big advantage when it comes to broad distribution of the product rapidly after manufacture and testing. I've been doing this for a decade. We have lots of data showing that this is a reliable vaccine platform to produce a full and reliable immune response. We have lots of success in this space with other viruses, specifically Ebola, and now with work that we're doing with Marburg. We're ready to go, and we're looking forward to having success here so that we can really make a dent in this awful pandemic.
Okay, thank you very much for playing that, Kevin. Before we get into this today, I want to thank everybody for joining us. We really appreciate your participation in today's Fireside Chat. I want to also remind you that there's a chat function on the Zoom toolbar, as well as a Q&A function, which you can use to put your questions in. We'll be taking questions in a little bit. So please, if something comes up, pop it into the chat or Q&A function, and we'll be able to address them. Second, thank you for tuning in today. This is the biggest crisis that we've faced over the last 100 years from a health perspective and on a global basis. I appreciate both of you guys taking the time and doing this with us today and sharing the information that you have. I've come to find that everything that you've been telling me since we first met is coming true, and we're seeing it play out throughout the world today, especially in the US. So thanks for joining us today, and let me jump into these questions. I know that you're initially testing your vaccine as a therapeutic. Can you elaborate on why you're taking this approach in Australia?
Sure. And Michael, I want to thank you very much for having us on the platform today to talk to the folks you work with. And I can't thank you enough for the opportunity to be here. So we need a therapy. We need a therapy for this virus. It's not enough to simply rely on vaccines. We'll talk about the problems with the vaccines that we're seeing today, but there are two big advantages to taking this approach. First, we need a therapy. And second, it's easier to test a therapeutic than a vaccine. Our drug is both a therapeutic and a vaccine, and by choosing to treat people that are infected with SARS early during the course of the infection, we can very quickly see if it's working, because we expect people to progress in about 14 days. And if we show in the active group that they don't progress, we can get data with a few hundred subjects instead of 10, 20, 30,000 subjects in a large 1 year long vaccine trial comparing unvaccinated to vaccinated people exposed to the risk of getting a viral infection. Why are we in Australia? I've done two phase 1 clinical trials there before in my previous biotech company. It's a great place to work. Regulatory wise, it's streamlined, and economically, the government is very supportive of biotech work, which is why many pharma companies are there from the United States and elsewhere. The phase 1 study is a study in healthy volunteers, and it's a required first step in clinical testing before we can test people that are infected with the virus. So by doing phase 1 data there, we can take that phase 1 data back to the FDA and other agencies and start a more expansive study of sick people throughout the world.
So Henry, when you have something you want to ask, if you can just pop in and
Okay.
follow on. Do you have anything that you would like to ask Reid to help him to elaborate further on that answer?
I think the only thing that you might want to add, Reid, is the quality of the people in Australia that also take a look at this to represent our interests in Australia and why this is safe.
Well, sure. So first of all, we're in Australia because we've had good success there before. We set up a company in Australia in 2018 specifically to be able to leverage the many advantages of doing clinical work there. There's great institutions there to work with. We're working in Perth with university folks out there, and also private sector research entities there. So we're well positioned to take advantage of this. The other reason we're very confident that we're going to have success here in this phase 1 safety study is that all of the chemical components in our vaccine platform are used in already FDA-approved vaccines and pharmaceuticals. So there's a lot of clinical data already on the safety of the basic building blocks, the microspheres that we use to encapsulate our product, and the chemicals we use to enhance the immune response, again, are already used widely in US pharmaceuticals and vaccines.
All right, excellent. Moving on to the next question. Reid, can you dispel fact from fiction on current vaccine technologies?
Sure, let's just start with the facts. I mean, these vaccines have been a remarkable way to give us breathing room here during a horrible pandemic, and everybody should get vaccinated. If you haven't been vaccinated, you finish the call, please run down to Rite Aid or CVS and get a vaccine. I personally think the mRNA vaccines are your best choice. Nothing really wrong with the J&J vaccine. I don't think it's quite as effective, but get any vaccine you can and get it right away, because in particular, the context of this deadly Delta variant, you really do not want to face that vaccine in a dark alley without protection, because it'll kill you. Are the vaccines perfectly effective? No, they're certainly better than nothing, but they represent, I think, a temporary stopgap, because we're seeing the vaccines experiencing breakthrough. Israel reported about 50% of their new infections are in vaccinated patients. Now, how bad is that? Well, you know, just because you didn't die when you got the infection doesn't necessarily mean you're out of the woods. We know that long haulers, people who have persistent symptoms, brain fog, blood clots, in some cases causing stroke or pulmonary embolism, cardiac inflammation causing cardiac problems. We don't know to what extent vaccinated people who get infected will or will not progress into those categories, because Delta hasn't been around long enough. So these are the issues you have to frame. And I think we have to just be thankful we have those vaccines today, but recognize we need a different approach to deal with the variants, because they're going to come wave after wave after wave. And eventually, my concern is they're going to completely break through the vaccine protection if we rely entirely on antibody vaccines. And the existing vaccines all produce an antibody response. And as we'll talk about, we need to be using the other arm of the immune system, the cellular arm, to have killer T cells attack, so that we can get rid of these variants effectively and really rid ourselves of this pandemic once and for all.
I've got one thing. Should you add at this time why some people are surprised that the Delta variant is so different from the first manifestation of COVID-19?
Well, this virus has just an insatiable ability to mutate and cause more and more trouble. And the Delta variant produces about 1,000 times more viral load than the original virus that we saw out of Wuhan. That's like a battering ram just banging against the vaccine front door. And eventually, hit it enough times with enough variants, we're going to see more problems. So I think that we're not used to seeing a virus pop up out of nowhere and be as stable as it was in man. It was stable in humans for about a year, and then it started to create these variants, perhaps because of the use of things like monoclonal antibodies in the UK, which may be why we saw the UK variant early. We don't know. But clearly, it can become more lethal and more infectious in ways that we're just not used to seeing in viruses in general. So I think everything the virus does is surprising. The brain effects are surprising. The blood clots are surprising. The cardiac inflammation is surprising. The rapid mutation, and of course we're seeing Delta become 100% of the viral infections in the US pretty soon. So everything about the virus is new and scary. And so when you have new scary things, I think it's critical to look out of the box at new solutions. And again, very thankful we have safe, effective vaccines today, but they're starting to weaken, and we have to quickly think about ways to continue to protect ourselves using different approaches.
So I mean, just one thing to clarify. So you're actually saying that when this particular virus comes under attack, it goes out of its way to become something new to evade attack?
The virus has, the coronavirus in general, have a proofreading mechanism that's not present, for example, in influenza, where the virus can be relatively certain that a mutation is viable, so that it's able to have a fit mutation where it won't just peter out. This is a very efficient tool for ensuring that it can create many new mutations, which of course by natural selection get to either survive or not survive depending on conditions such as vaccines or therapeutics that are in the environment. So it's a very clever virus. And this is part of the reason it's scary. Scott Burkholz is with us in the call with Henry, is head of our bioinformatics group. He published an article with our collaborators at Stanford in a prestigious journal called Nature just a couple of months ago, really describing how many ways spike can and will continue to mutate into these fit structures that are viable for the virus, so they can keep coming at us again and again and again with new variants. And my concern, as I mentioned before, is that that's going to result in a complete breakthrough of the virus through our current vaccine technologies and make antibody vaccines useless. And this is why we have to sound the alarm here and realize, even though we're kind of doing okay right now, although let's not forget, people that are vaccinated to get Delta, 0.5% of them die. So every million people that get infected were vaccinated, 5,000 people die. That's a lot of people. I mean, 0.5% doesn't sound like a lot, but if you're talking about half the vaccinated folks getting infected, it's a significant Grim Reaper event happening there. So, but we really have to really focus here on other solutions.
All right. Thank you very much for that answer. Dr. Rubsamen, can you help us to understand the current state of coronavirus outbreaks in the US and globally? Can you go into some detail?
So what happens? So this virus pops out of nowhere from Wuhan, China. I was actually volunteering at a rural trauma center in southern Michigan, Christmas of 2019, before really the outbreak became a real thing. And we had a patient come in with a huge pulmonary embolism, big blood clot. We had the Tropicos catheters down the neck veins to use ultrasound to lyse these clots, and we saved his life, thank God, doing that. But in retrospect, he probably was a COVID-infected patient. I went back several months later to get blood samples to do antibody assays, but we didn't have any frozen samples to test. But that's what's amazing about the original form of this virus is it was stable for a year. That's unusual. A virus that lives in an animal population like bats, you typically expect to behave like SARS-1, kind of comes and goes. This thing was solid and stable for a year in humans and spread globally right away. This may be because it was produced by gain of function research in Wuhan, China, which means that it's possible that researchers exposed it to human lung tissue to make it more stable, at least initially, in a human infection context. That's not proven. I know there's some controversy about that, but that actually does explain in sort of the simplest way why we're seeing this phenomenon. And then once the virus sees selection pressure, such as antibody therapy, which may be why we saw the alpha, we use names for these variants that don't mention the country. So the UK variant is the alpha variant, or who can remember the codes? I guess the B117. I mean, but there was antibody therapy testing at that time. Maybe that was selection pressure that caused the alpha variant. We don't know. Now we're seeing, fast forward to the Delta variant, 1,000 times more viral load measurable in the nose. That really is a battering ram. If you think about it, more viral load, more opportunity for infection to occur in the host. So the ability of the virus to mutate has enabled these variants to occur. And although we see kind of a relatively quiescent period right now in the US, that Delta variant is really wreaking havoc in India. And of course, the Lambda variant you've heard about is wreaking havoc in Latin America. So these things are out there in their current state causing problems because this virus can mutate so efficiently. And again, my fear is that we're gonna really be in trouble here when we get a new wave, a new variant wave, next variant, 10 variants from now, 5, I don't know how many variants from now, but this thing could hit us with a completely resistant variant that the vaccines are completely impotent to deal with, which of course means the antibody therapies won't work either. And this is going to create a situation where we're in very serious trouble. We don't want to be developing a solution from scratch at that moment. That'll put us back into a 2019 type scenario. We'll be talking about, God forbid, lockdowns, economic disruption, larger death than we saw before. This really is scary to me. And this, again, is why we have to be thinking so clearly about what can we do differently.
I guess the only other thing that you could add is, under the surface, people in the know really do believe that this, although we're probably never going to be able to prove this from China, people really do believe that this was engineered in a lab.
Well, there's an old saying in science called Occam's razor, meaning what is the simplest explanation
that kind of puts everything in perspective. You're absolutely right, Henry. We're not going to be able to prove this. But when you see a virus this stable for a whole year after a first jump from animals to humans, you have to start asking questions like, why is this happening? A simple explanation, since we know gain of function research on other viruses was happening in Wuhan, is that they don't. Now, why did they do that? It doesn't mean they were, Machiavellian crazy scientists. It means they wanted to see, gee, we have this virus. What could it do, and how can we develop therapies for it? It's a research program that was carried out around the world for many years. There was a pause on it from 2014 to 2017 because researchers said maybe this is too dangerous to do, but there are reasons why you'd want to do it, and I don't think you can impeach people for wanting to do that, to study new therapeutics. But if such a work was being done and it leaked out of the lab, then you start understanding more easily how this thing could have appeared out of nowhere with excellent binding characteristics to the ACE2 receptor, which of course is how spike protein helps the virus enter cells, and how stable this virus was for a year and how able it was to spread so widely.
Dr. Rubsamen, can you help us to understand the strengths and weaknesses of the mRNA vaccine platforms?
Sure. So big strength. They were rallied really quickly. So we had in this country the availability of good vaccines against this virus made very quickly. Pfizer and Mrna, we perhaps really have to be off to those companies and to the way they were funded so quickly by Operation Warp Speed to do this work. And they produce an immune response. They basically cause your body to manufacture, in the case of Pfizer, the whole spike protein. In the case of Mrna, the receptor binding domain, which is a part of the spike protein that binds to this ACE2 receptor, so that your body can make antibodies to these targets and neutralize the virus. And those antibodies were, in fact, neutralizing early on during the epidemic and provided us with protection. So that's good. The weakness really is that they are antibody platforms. And again, antibody vaccines, they've been working great for many, many years. Polio vaccine, measles, mumps, rubella vaccine. There are all kinds of hepatitis B. There are all kinds of vaccines out there that produce antibody responses that work great. But there's no vaccine for the common cold. You can imagine there'd be a lot of money in such a vaccine, but common cold's a coronavirus, and coronavirus is tricky. We've always known that it's tricky. And so, thank God we had enough tools to deal with this pandemic, even though we didn't have a lot of experience dealing with coronavirus with a vaccine technology, but it's great that they were there. The problem is, and this is a somewhat simplified explanation, but antibodies pretty much can only see the outside of the virus. And the problem is that is where it is mutating most efficiently. The spike protein, which is this thing that interfaces to the ACE2 receptor, which is on many different kinds of cells in our body, including the nervous system and blood vessels, that's where the mutation is happening. So that's the fastest changing target. If you go inside the virus, where this thing called nucleocapsid lives, that's not mutating nearly to the same extent as spike. And in fact, we were intrigued, and we've done work on this for some time. We saw the same kind of phenomenon with Ebola. Survivors of the 2013 Ebola epidemic had circulating killer T cells attacking this nucleocapsid protein. So we took those same targets that were favored by these survivors, and we put them into a mouse vaccine. We tested it in mice at the US National Lab at Galveston, 100% protection against Ebola with a single dose. So we published that in a peer-reviewed journal article, and we actually speculated in the article, gee, this was during the pandemic. We said, gosh, we wrote the article during the pandemic and said, maybe we could make a coronavirus vaccine using the same strategy because it also contains this nucleocapsid protein. Now, we had previously gone to the US Army with our Ebola data a couple of years ago. We got an Army contract to develop a Marburg vaccine, which we designed under a US Army grant, and which we're now manufacturing for testing in primates. The Marburg is a weaponized version of Ebola, using the same basic approach. So we developed this anti-nucleocapsid SARS coronavirus vaccine, and we went back to the US National Lab at Galveston, one of the few places you could study these deadly viruses at. And we gave the vaccine to primates. We used an inhalational approach to give the vaccine to the primates, and we protected the primates against pneumonia, and we published that result in a peer-reviewed journal article just last May. So we really feel like we have a have demonstrated that the cellular arm of the immune system, the arm that produces killer T cell attack, can attack at the center of this virus, can attack this nucleocapsid, which is a sitting duck. It's sitting there quiescently, not mutating very much at all. And why is that? Because it's critical to viral replication. It can't change very much or the virus would die. And we pick targets on that nucleocapsid that are also present on SARS-1 to make ourselves even more confident that they won't mutate. So we feel like we have a winner here, and primates have an immune system very close to that of humans. And, I'm very confident we give this by inhalation to humans that will show a safe and effective immune response, and later in sick people that we can reverse the infection, hopefully prevent anybody from becoming a long hauler, and possibly even treat long haulers who may harbor the virus. So we think that this killer T cell approach is critical. And as I said in the video, I don't think there's anyone else in the world with a T cell attack model, T cell attack vaccine therapy that's been tested in primates, demonstrating effectiveness. So we're very excited about this, and we think that we can really move forward and make a dent in this thing.
Reid, please explain why the variants are going to continue and are expected to be more easily spread as well as more lethal.
Well, as I alluded to, Scott Burkholz, again, he's head of our bioinformatics group. We have great collaborators at Stanford. We published 2 articles with them already. And one of the articles, again, was in Nature recently. And this article is a very elegant article, really saying, hey, this spike thing has enormous opportunity to create variants. There are many, many positions in spike protein that can change, can mutate, that are viable, meaning the virus has all kinds of cards in the deck. It can just keep it can just keep hitting this at the blackjack table with those cards again and again and again before it runs out of gas. So I think we really, this is a roulette wheel that's never stopping 24 hours a day. When it comes up 00, we're going to be in serious trouble. And it's only a matter of time, really, I think, before that happens. If the virus had a lot of dead ends, a lot of mutations that would kill it, maybe I wouldn't be so concerned. But this thing has an enormous capacity to continue to produce variants we can't even conceptualize right now. So you're going to see more variants. You're going to see them. And again, the Delta went from, 0% to going on 100% in a matter of months. This thing produces variants much faster than we can make boosters. You haven't even seen the first booster shot yet from the mRNA companies, and we're already on multiple variants. So the worry is we're going to get a variant that's going to blast through the vaccines. It'll be 2019 again. We're going to be in serious trouble. I think this is almost a certainty. It's depressing to think about that. It's depressing to say it, but I think we have to be wide open here. I really do. Again, just to get back, I don't want to belabor myself here, but I'm a practicing physician. I work in the operating room 8 days a week, 8 days a month. Sorry. I enjoy teaching residents. It's really my calling. My whole work in biotech is just to provide more tools, take better care of my patients. That's really why I do it. We're going to need this tool. We're going to need it. We're going to need it in quantity, and we're going to have to develop it fast, because I don't think we have a lot of breathing room here. We're lucky that the Delta variant isn't completely breaking through this thing.
So let me interrupt you for 1 second.
Sure.
You're not the only person that knows this. Scott's not the only person that knows this. There is a consensus underneath the surface of things that really is aware of this, right?
Sure. So I think that those of us in the front line who continue to see our patients get sick in the context of the virus are worried. But look, I mean, nobody, we just all took our masks off. People go to the restaurant. Nobody wants to say the glass is half empty. Look, I'm completely sympathetic to this. I mean, it's depressing to think about. This is not a fun thing to think about. This is depressing. And it's depressing politically, it's depressing economically, it's depressing personally. It really is. My wife's over at Lake Tahoe this week, and I said, honey, eat outdoors and eat at home, nobody wants to be living their whole life constrained by a bogey that you can't see right in front of you. I'm just saying that the scientists who work on this many of them share this view. And I think we just have to realize that the data is pointing in a pessimistic direction. And if you're a scientist and you see the data, gosh, I think you really have to be concerned. And I think solutions, and by the way, I'm agnostic to who the solution comes from. If someone else is a great solution, I mean, I read literature every day, and I think I don't relish being at the tip of the spear here. Believe me, I'm not, oh gee, it's just fantastic. We're the only people with this solution. It's actually terrifying. But because who wants that kind of responsibility? But here we are with this advanced development thing that seems to work really well, and we need tools to be able to make more of it. We're very fortunate to have a manufacturer right here in Cleveland, half mile from my house, Oakwood Labs, that's already manufactured samples for human testing. Very fortunate to have Lockheed Martin, Aerospace Skunk Works working with us to optimize our high volume manufacturing dry powder systems to make them able to make lots of material. Very fortunate to GEA, German company, to sell us a very large capacity piece of equipment. Very fortunate to have College Station, Texas, Operation Warp Speed, happy to work with us to make more of this stuff. Not Warp Speed, the government, but the technical people that make stuff for Warp Speed. All that's great, but it actually scares me to think that we may have the only solution. I mean, it's great that we have a solution. I'd rather not be alone. I'd rather have 8 companies doing this, but we're here. We're here by ourselves with a solution that I'm happy to tell you about today, but it's scary and it's thought-provoking and it requires focus. So again, we're here and we're trying to move this forward so that we can make a dent in this horrible pandemic.
Doc, as somebody who's had, I was hospitalized in November, for those of you that don't know, from coronavirus, and I spent 21 days in the ICU. So I understand what it's like to be a long hauler. Why has recovery from COVID been less than perfect for so many of us, recovered COVID victims? And why is there a need for therapeutic that'll arrest the development in someone just testing positive?
The NIH has devoted almost all of its COVID budget and much of its regular budget to understanding this question. What is long hauler? What causes it? What do we do about it? So really, we're just in the phase now of characterizing what it is. And what we know so far is at least 30% of people who get a COVID infection become chronically ill. And in many cases, this becomes a disability, requiring time off work, requiring access to expensive healthcare, and of course, a lot of personal suffering. What characteristics are there of long hauler syndrome? Well, I mentioned this before. Neurological problems, brain fog, blood clots causing catastrophic strokes, very large blood clots in very large vessels, which we don't see spontaneously occurring often in healthy people. And we're seeing this in people infected with COVID. Cardiac inflammation, making the heart angry and red and sick so that it doesn't beat properly or has horrible dysrhythmias or a combination of all the above. So these are just some of the things we're seeing in this long hauler syndrome. Is it due to persistent viral reservoirs? Well, if it is, we should be able to actually help long haulers after the fact, because killer T cells can kill infected cells. But I think also equally as importantly, I believe people who get acutely infected, even if they're not immediately sick, we should arrest the virus in its tracks. If you get a COVID infection, virus infection, whether you're vaccinated or not, I would want to give you a therapeutic, for example, what we're developing, to arrest it, to drop the viral load to zero and stop it from progressing, because only in that way can you perhaps prevent this long hauler syndrome. So prevention, an ounce of prevention is worth a pound of cure. I'd rather clear the virus today and not have long hauler occur than play from a losing position. Even though I think we might be able to help long haulers, I feel very confident if we got this virus and nipped it in the bud by treating all acutely infected people, we could prevent it from happening.
Henry, do you have anything you want to add here?
I guess what you're saying, Reid, is it's not just enough to feel more secure about not dying because you've been vaccinated. That there are other problems from getting it.
Well, I'm concerned. I am concerned, and again, we're operating from some ignorance here, but I'm concerned in the press that if you're vaccinated, you get Delta, you won't get too sick. Well, yeah. Of course, we just said 0.5% of the people end up dying, but okay. But we don't know today if just getting a little sick does or doesn't cause you to progress into this long hauler mode, because Delta hasn't been around long enough. So will you get blood clots? Will you get cardiac inflammation? Will you get brain fog? Well, maybe you will, maybe you won't. The data isn't in yet, but it would scare the heck out of me if I got the infection as a vaccinated individual. And so, you know, I think we have to be very conservative here in how we think about this, and very aggressive about how we think about what we're going to do going forward to prevent this stuff from happening.
So when I had the opportunity to get the vaccine, I took it immediately. But this next question goes into some of the alternatives to the current antibody platform-based vaccines. What are the best or better alternatives that are available today?
Well, as we mentioned, so the body has two arms of the immune system: the antibody arm, which is also called the humoral arm, and the cellular arm, which involves killer T cells that kill infected cells rather than neutralizing antibodies that bind the virus directly. As we discussed, these antibodies pretty much can only target surface targets of the virus. That's where those are bad targets, because spike, which is on the surface, is mutating very efficiently. Inside the virus is this very important protein called nucleocapsid, which is not mutating. We know from the literature, survivors of the SARS-1 epidemic have circulating T cells 17 years later that are targeting nucleocapsid. There's great literature on this. So not only is the targeting probably better because it's a non-mutating target, but the longevity is much longer. You're talking about 6 months to a year for antibodies, maybe. You're talking about years and years for killer T cells. So the cellular arm of the immune system provides a very interesting alternative to the antibody arm. Could they be used together? Maybe. But I think you have to have the cellular arm active and operational if you're going to really take a crack at the variants, because the variants all have something in common. They have a stable nucleocapsid protein that's sitting there with very little mutation. And we follow this mutation very carefully, and it's not mutating in ways that will interfere with our targeting that we're doing with our vaccine and therapy. Same product, same drug. So I think that it's critical to explore a killer T cell vaccine therapeutics. It's critical to understand that the spike protein could be a bit of a dead end as it keeps mutating. It could become ineffective. And so the alternative to the current antibody platform is a cellular T cell vaccine platform. Our vaccine doesn't contain cells. It contains little tiny antigens. I just saw a Q&A just flash by. What are the components of the vaccine? Well, it's microspheres that are made of a biodegradable polymer called PLGA that's widely used in the pharmaceutical industry, the same size about as a white blood cell. And we put in there the target peptides, the 3 peptides that are sequences that represent unique signatures, if you will, almost like a digital serial number on the nucleocapsid protein that are constant. And we put in the sphere a synthetic little piece of DNA called CPG, which is a artificial bacterial DNA. This is used in a marketed vaccine in the US, and it makes the body say, oh, I just got infected. I better pay attention to this microsphere. What's in there? So it's a Trojan horse. It triggers the immune system to seriously consider these tiny, tiny peptides, which normally would go unrecognized, and causes killer T cells to increase in number and go out and attack anything that expresses those targets. And of course, what expresses them is the infected cell. The cell that's infected with SARS will have all those nucleocapsid targets on the outside of the cell. Killer T cells will see that and kill them. We have a diluent, that's a dry powder that contains the microspheres. We have a diluent that contains water and a thing called MPLA, which is monophosphoric lipoic acid, which is bacterial cell membrane analog. This is also used in one of the cervical cancer vaccines marketed around the world. And that also makes the body say, hey, I'd better pay attention to what this person just got. I'd better look at it. It's important. And that level of importance assigned to our vaccine therapeutic by the immune system is what ensures a solid immune response. And again, all those components, PLGA, MPLA, CPG, are all used in marketed FDA-approved vaccines and pharmaceuticals. Their safety profile is well characterized. We could have used they're called adjuvants, other mechanisms to enhance the immune response. We did not choose those even though they might be pretty interesting because they're not an FDA approved products. As a physician, I don't know, I just cannot, I cannot take the risk with any subjects in our study. I want this to be really absolutely safe. We're not doing anything edgy here. I don't want to say, well, we'll just do edgy stuff. It'll work better. No, we're not doing that. We're not doing that. We go for safety first. The first, second, and third thing in the design is safety, safety, safety. Then we worry about, will it protect, will it work? Because we're not going to sacrifice patient safety to make, quote unquote, a better therapeutic. It's a bad way to think. I put needles and drugs in patients 8 days a week. It's a very sacred, really, trust that patients and study subjects have in us as medical professionals to take care of them. And we're going to take care of them like one of our own, like our family. And we're not going to take any risks. So really, we took a very conservative approach here with the design. So thank you for that question. I just glanced at that. We'll get some more questions later, but please go on, Michael.
Absolutely. Dr. Rubsamen, why did Big Pharma choose to go down this path? Why did they choose what they chose for all of us?
We're wrapping up the way we started, and that's to say this. I mean, of course, Big Pharma had to move fast. I mean, you got the government saying, we're in deep trouble. Here's a bunch of money. Give us something fast. You're going to take what's on the shelf. Mrna, Pfizer, J&J, they had technologies they developed for years. They knew how to do this. They weren't going to invent something new. They were going to suggest to do things. They're going to go out there and do whatever they could to get us the best possible therapy quickly. And those three companies should get a big pat on the back, in my view, for really leaning into this thing with a lot of unknowns and just making it happen and dosing patients quickly. I think Mrna had the first phase 1 patient study with some mouse data and just did it quickly. More power to them. It was great. But of course, they didn't have time to sit down and have the conversation we're having, which is, okay, that was good. How do we deal with sort of the next phase of this thing? So we're sitting here saying, okay, we got some breathing room. You can get the vaccine. It provides significant protection, but the variants are a battering ram on the front door. You can hear it banging away every time you look at this data, and it's going to break the door down. There's going to be a variant that's going to break through these vaccines, and we're going to be in big trouble very quickly. And it'd be a tragedy if we weren't using all of our brain power and person power and everything else to say, okay, what are we going to do when this really big bad thing comes out to get us? And the answer, in my view, is you go to the cellular arm of the immune system. We know that people who survive SARS-1 have T cells, killer T cells attacking nucleocapsid. We know that was same with Ebola. We know we made an Ebola vaccine against nucleocapsid that worked in animals. We have a SARS-CoV-2 COVID vaccine that worked in primates against nucleocapsid. It uses all ingredients previously approved. It should work in people. We're trying to test it in people now. I mean, really, this is what we all have to be thinking. I invite others to think the same way. I'm not trying to carve out some kind of empire here. We're just trying to get our solution out there quickly because it's across the street from my house. I've got 800 vials sitting in a plant ready to clear stability in September, ready to test. So we really want to get this out there right away so that we can demonstrate human data. And I guess we created Strategic Vaccines as a public-private partnership because that's a vehicle that's really important if you want to get government financing. We are we've funded this company with private financing so far. My biotech Flow Pharma has funded all the Strategic's work so far. And fundamentally, we're going to need government funding to do this and roll this out in a big way because it's obviously critical to do that, to do rapid expansion. But you cannot, interestingly, you cannot get a large government scale-up grant today, and this was true during the first phase of the epidemic as well, without some human clinical data. The first line, blah, 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 opportunity, apply here, blah, 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 must have human clinical data. Well, the must have part means if you have primate data, that's not good enough. So we need to get some human data quickly, some human data quickly. These applications don't even require a lot of data, but some data to show that, yes, you put it in a person, you got a safe immune response. And we also want to move quickly into at least a small study to say, okay, and if you have the virus, we can shut it down. So this is the kind of data we need to get and unfortunately, it's a lot of money, but you have to be able to make a lot of this. It'd be heartbreaking to me if we show this works, after we show that it works, we can't make a lot of it quickly, because then, then it's, then it gets really frustrating. So we've actually invested quite a bit of time and money already in understanding and beginning to set up the systems to make a lot of this fast. We can already make it today here in Solon, Ohio. We can make quite a bit of it, but after this is proved efficacious, we're going to make a lot more. So the Operation Warp Speed folks, the technical people there are very interested in helping us. We've been working with them for a couple of years on setting the stage for this, and really since the beginning of the outbreak. And we're very grateful to their help. Lockheed Martin Skunk Works has been great to work with again for the last going on 2 years now, to help us use their computational fluid dynamics modeling to look at commercial systems that we're specifying and modifying them to optimize the production of our microspheres that make this vaccine. Oakwood Laboratories across the street specializes in microspheres. So here I am 2 years ago. We're working on this Marburg program for the Army. The Army says, well, gosh, we're working on COVID, so we can't work on your project with you for the next several months. I said, well, we'll take our resources internally and make a COVID vaccine. Well, we didn't have any money to fund manufacturing of this stuff. So I go over to Oakwood, which I've been talking to for a couple of years about other projects. I said, look, you guys, we have this COVID epidemic. We don't have enough money to pay you to make any of it. We can buy the raw materials for you. And we gave them some stock in our company, and gosh, they leaned into it with, they had something like 20 people working on this full time, and they made the vaccine for us quickly. And I'll just give a plug for the Midwest. I'm from San Francisco. I've lived in Ohio now for almost a year, and I've been coming here working with Oakwood and with collaborators, scientific collaborators over at Case Western Medical School, where I'm on the faculty. They're teaching the residents, as I mentioned. But, I'll give a plug for the Midwest. In the Midwest, it's okay to want to save humanity, right? You don't have to start with the billions of dollars you're going to make. It's okay to say, we are going to work really, really, really hard for the public good. And of course, if this is successful, it's a big opportunity. Yes, there's plenty of money available, but you can open with, we're just going to lean into this to do the right thing. And I'm really proud of that. And I'm proud to I was accepted, I think, very quickly into this great community here in Cleveland. Henry's in Shaker Heights. We became good friends. Actually, Liz Evans at the law firm introduced me to Henry because her father was a well-known lawyer here. I also want to thank K&L Gates, our law firm, for introducing us to Henry. Also introduced to Lockheed Martin and folks that helped us with the Warp Speed project. Their public policy team has been great, also helping us field applications to the federal government, some of which are still pending. So we've been very fortunate to have a lot of people stepping up big, way beyond really exclusively any kind of business interest, but to really see the need here, what we can do. And like I said, I'm a physician first. I still practice 8 days a week at the hospital. I really enjoy that. I enjoy meeting patients, helping them, teaching the residents. And I quiver a little bit as I walk through the ICUs. I see, of course, fewer and fewer patients with COVID, but we just need to be ready for the next big wave. And I think it is coming. It is coming. And thank you very much for listening to this talk. We'll go through some of these questions now, the Q&A, but thank you again. Michael, shall I just run through some of these questions?
Well, there's a couple of things that came up that I wanted to ask you. As I've shared with you before, I'm still personally dealing with the issues from having COVID, and my family's still dealing with the issues from COVID. It's very serious. It's very stressful. It's traumatic. It's traumatic on an entire family. It's traumatic on the person who gets it and finds themselves isolated and facing near death. But to me, what's coming up through the conversation is a thought, just a question. Why do you believe that you're the right person? Why do you believe that the team at Strategic Vaccines is the right team? to produce a breakthrough vaccine and therapeutic, really for humanity.
Well, there's an interesting comment people make now about biotech, and that you really can't do any biotech now without bioinformatics, data science, and healthcare medicine type knowledge. So you need two kinds of people to come, people that understand the sort of medicine immunology part of the equation, and people that understand the very complex computational tools that are required to really navigate through the genome, which is what you have to do to design something like this. We didn't talk about this in detail, but when you design a T-cell vaccine, you have to consider the HLA type, which is really the serial number of your immune system, and understand how to make it so people all over the world can respond to it, because a T-cell cares about your HLA type. A antibody does not. So what we did was we used bioinformatics, artificial intelligence, to not only pick targets on the nucleocapsid protein that we predicted would be very least likely to mutate, we also used the same techniques to understand what targets to put in the vaccine that would ensure that almost everyone on the planet would recognize our vaccine and make an immune response to it. So we are very fortunate, as I mentioned, Scott Burkholz. We have a number of folks on our bioinformatics team, Rick Harbach, Lou Wang, and others. And we really were able to attract the best and the brightest to do this. We have people working on the team that have a lot of experience in inhalational drug delivery. I was in that business for 10 years. I worked to develop one of the first inhaled insulin products ever tested, for example. And so we have background in the kind of formulation work we have to do. We're able to attract great talent in bioinformatics. And by the way, all the guys on my team can mine Bitcoin. So they could be doing that instead of this. So they're here helping us because they believe in the humanitarian objective here more than mining Bitcoin 24/7. So I appreciate, their dedication to this, given the options they have to work in a very competitive environment and do work for tech companies involving fintech and so on. So I think that a lot of it's luck in attracting the right people, but it's all about the people. You can have a great lab. We have a great lab. You can have great tools. We have great tools, but it's really the people that are the heart and soul of the company. And not only people that work on the inside, but the great folks at Lockheed Martin Skunk Works who've solved huge problems for our military, including making the first stealth aircraft systems. And these are people who think out of the box. They're 100% into this program. The guys at Operation Warp Speed who enjoy what we're doing at the Seattle Group, which is the group at Texas A&M that does the manufacturing for that program. We have a public-private partnership with them and our strategic vaccines company. These are just wonderful people. So we've been able to attract the best people to help us. And I just can't thank those people enough. And also we've been very diligent and just working on the T-cell platform for 10 years. And really by just keeping, develop something, publish a paper, develop something else, publish a paper. We always develop and publish, develop and publish. We have a paper trail of peer-reviewed journal articles, all indexed with the NIH Library of Medicine. We really try to publish as we go so we can be very transparent about what we do. People can read what we do in the literature. They can help us understand it, learn from what we're doing, because we want to be very collaborative in what we're doing. So I hope that answers your question, but that's kind of how we got here.
It does. But before we take Q&A directly from the audience, I want to address what Strategic Vaccines needs to combat the greatest humanitarian crisis of our time. And I'm asking everybody that's listening to carefully consider learning more about what Strategic Vaccines needs, right? This is not very serious until you can't breathe or a family member is dying. So all the work that goes into building this company, it's not free. The company needs some stuff. And I want to invite Henry to briefly address what Strategic Vaccines needs.
Okay, well, I'll do my best to be brief, which is not my nature, but I will. In October, I was contacted by Liz Evans, who is the daughter of a very close friend of mine who is one of the most prominent litigators in Cleveland, Neil Evans. And so Liz asked me if I would help Strategic Vaccines raise $25 million. Based on how I have always approached making recommendations in my different financial businesses, I had to do some due diligence. My son happens to be a neuro radiologist with boards, and he was very skeptical how something could be called a therapeutic when it was really a vaccine. And I started reading some stuff, and then I know a few people on the staff at Case Western Reserve Medical School real well, and I did some due diligence. And so I decided that what they were doing from the viewpoint of others made sense. And then, of course, I got to know Reid. And when I get involved with people, it has to be based on integrity and the character, apart from just the idea of whether something is going to be successful financially. And so I, over the last 8 months, have grown to know the people that are dedicated to doing this, and the motivation. The ulterior motive is to save lives. There really is no other motive for what they're doing. And everybody knows there is an ability to make a lot of money if something works in biotech, but the real motivation here is the character and the devotion of everybody to making this work and saving lives. And so I'm really proud to be a part of this. And the $25 million I restructured when I got into this so that 12 million is due for the first phase of testing. And then the second 13 million is not due until FDA approves going forward with phase 2, which means it's been proven safe. So that mitigates a lot of risk. It just reduces a lot of risk. And then my other feeling, and the way I've always approached making recommendations, is the more you know about the substance, the material substance in place, the more predictable the future success will be for whatever you're investing in. I know from my venture capital experience, and this is not a normal venture capital ask. It's about a 10 bagger or an 8 bagger, and then people fire birdshot into the air and assuming that the numbers will work out, that you're going to hit 1 out of 10 and you're going to make enough money out of 1 to pay for all the losses. This is not that kind of a situation. So really, this is about asking people to join Strategic Vaccines in the mission to save lives and to bring something that is really, really necessary for the future of the health of the world into being. And that's why I got involved, and that's why I'm more involved. And so my role has basically changed a little bit from the beginning. And I'm just really proud to be a part of this, and lucky to know a guy like Mike Corselli too, and all the other people who are involved in this who really care about doing the right thing. And of course we have financial projections which can be backed up by facts, which I believe are incredibly prudent, and all kinds of other information to share with people who are interested in joining this mission. And one thing for sure is everybody will be totally transparent. Everybody will tell you everything they know, and there are no bad questions. And even at the NDA requirement that is the norm has been removed for most of the information in the K&L Gates data room. And of course, you have to have an NDA for a couple of Lockheed contracts and some contracts with the government. But other than that, everything is basically transparent. And another important thing to know is the law firm that's part of this is one of the 10 most important firms in the world. They have a very well-established, very highly respected group of lobbyists in Washington, D.C., which can help make the public part of this funding real. And that also doesn't dilute the money that's in there that comes in now. So I think this thing is structured very well. The term sheet is extremely favorable for the investors because I have spent my life representing investors, and everybody in management is very concerned with being transparent and being on the side of the investors. So, I mean, that's where I'm coming from, Michael. And so I guess I'm basically in charge of talking with people who are interested investors and then making certain that they helping facilitate their due diligence.
Shall I go through some of these Q&A questions, Michael?
Yeah, let's do that. But I just wanted, we always start these and end these on time. So For those of you that joined that are going to be dropping off now at 11:30, I want to thank you for joining. Let you know that we'll be sending out a recording of today's presentation, as well as a transcription of the Q&A. For those of you that want to stick around and continue to ask questions, we're going to get into the Q&A session right now. And lastly, before you drop off, I am making a strong emotional appeal. It's because I'm suffering from PTSD from what me and my family went through last year. But for those of you that don't have a heart by now, after what we've all been through over the last year and a half, I encourage you to get into the data room and check out the financial opportunity that's available. I almost feel embarrassed saying that, but it is a financial decision and you should consider it. So why don't we jump into the Q&A? We got a ton of questions. We got some stuff in the chat.
I'll go to the Q&A, and I'm going to try to answer all these questions quickly. I want to thank everyone for asking the questions. First question, your screen is very blurry. Sorry. I have a blurred background. I hope I'm not too blurry. What are the ingredients of your vaccine? We discussed that. We have this biodegradable microsphere, polylactic glycolic acid, one of the most widely used biodegradable products in pharmaceuticals. We have 2 adjuvants that promote the immune response, monophosphoric lipoic acid, or MPLA, which is a synthetic bacterial cell membrane, and CpG, which is a small DNA fragment that simulates bacterial DNA that makes the body say, oh, I just got something important. I may be infected. What is in there that causes the immune system to dissect our microspheres, look at the peptides inside, and teach T cells to go after those specific targets all over the body. So those killer T cells will propagate and have a sensor on them that matches the peptide and kill any cell that expresses that target. Did Delta variant have a long-lasting impact in India? It seems to have significantly deteriorated. In May, they had about 400,000 daily cases. They're down about 90% to 40,000. So clearly they're having fewer infection rate. It laid waste. It killed essentially as many people in India as it killed in the United States. I think it killed about 400,000 in India. That's, I guess, a little more than that here, but it was a big deal in India. And of course, it's 80+%, 90%, approaching 90% here in the United States. So these things kind of make their way across the world and do damage. And as I said before, if you're vaccinated, obviously the death rate is lower. It's still about 0.5%. But we need to worry about a down the road variant that would be much more devastating even to vaccinated people. Do you think this virus was engineered? I personally think, personal opinion, can't prove it, that the Chinese were doing gain of function research. That we know they were doing because it was funded and by the NIH. It was not with this virus, with other viruses, but they had the tools to do that. Gain of function research is important because you'd like to know in a closed lab setting what lethal consequences could occur if a virus mutates. You can go to work early on therapies. It's a noble proceeding if you have the right tools to do it. And it may in fact have been that the virus then escaped out of the lab to become obviously a problem. What about immunity? Go ahead.
Excuse me, can I just jump in for one thing?
Of course.
Okay. It is known that there was a researcher from that lab who mysteriously died shortly after blowing a whistle, called a whistleblower. That's been reported in the news. There also happens to be a younger woman who is an MD and also has a PhD in virology, and who has sought refuge in the United States, who escaped from China, who also has been speaking about this a great deal as a whistleblower, who is really has no other reason for saying this because she's put her life at risk, and she's still out there today. So we do have some whistleblower reports about this, and and possibly we'll never prove it because I can't believe the Chinese are going to just open the lap up and say, hey, we did this. But nobody is saying that it was done on purpose.
I think also it's Occam's razor again. It does explain a lot. If they were doing gain of function research, for example, passing the virus to human lung tissue samples, it would explain why the virus was so stable and so effective as a virus in human populations right after it jumped from animals. So it does explain things, which is why I think a lot of us like the theory based on what we're seeing. What about immunity after the virus? Not as good as immunity from the vaccines. We know this now. If you had the infection, you do want to get the vaccine. Immunity after the virus may have some benefit, but we know the mRNA vaccines in particular are better than immunity from natural infection. How do you explain that there are people who have seemed to be exposed many, many times in different ways, getting uninfected? This is a very important question. And so I didn't get into this, but there are folks who were exposed to SARS-1 who naturally, on their own, created T-cell responses to nucleocapsid. We know that because people have drawn their blood and published papers about it. People's immune systems aren't completely dumb, and they can spontaneously mount an excellent immune response to a virus. The purpose of the vaccine is to get that to happen optimally. And in the case of T cells, you really want to guide the patient's T cells to the sweet spot, to the targets that are not mutating. Left to its own devices, the immune system will typically not try everything that it can. It's not a shotgun. It fires a couple of rifle shots and then quits. Why? There's a phenomenon called immunodominance, where it kind of piles onto the first thing that looks interesting, even if that target is easily mutated away from the attack point. So it's very important to train the killer T cells to go to a sweet spot. If you don't do that, most people will not naturally hit the right target, but some will. And those people who you're talking about are people who probably mounted an effective immune response themselves and may have excellent long-lasting immunity. Are you saying that 50% of vaccinated people could die if they get Delta? Could die, but we know from the data that about 0.5%, about 0.45% of people who are vaccinated that get Delta are dying. That's about 5,000 per million. That's consistent with the Israeli experience. So certainly not all of them, but that's 5,000 people that you'd like to not die if they got a vaccine, of course, that was fully efficacious. The second thing I'm worried about isn't just dying from the infection if you're vaccinated. Will you become a long hauler? Will you get blood clots, brain fog? Michael talked about PTSD, of course, and cardiac problems. Will you get those things? And we don't know yet because Delta hasn't been around long enough. There's lack of credibility from the US CDC. Look, am I a politician? No. Am I a government worker? No. Do I know anything about it? Almost nothing. But obviously, these guys have a job to do. They have a complex environment. They have many bosses. I have no idea who's telling them to do what. And I don't even feel like being critical because there's probably no way I could ever work there. So I don't know enough about it. So, but I'm not quick to throw people under the bus because the phone's ringing from 5 different people. You do what you can. So I don't know. What about long-term safety for your mRNA vaccine? Of course, we're not doing an mRNA vaccine. I probably should have made that more clear. We're doing a peptide vaccine, meaning that we put these peptides, 9 amino acids long, very, very short, in a microsphere. And those peptides are made synthetically by a machine, but they precisely match targets on nucleocapsid, this non-mutating protein, that aren't mutating. So that teaches, programs, tells, causes killer T cells to increase in number and circulate throughout the body like sentries with maps and rifles to find the target and kill it, i.e. an infected cell and kill it, which is why we could use this as a therapeutic as well as a vaccine. And it says about safety. Well, we're picking the platform chemicals, the microsphere material, polylactic glycolic acid, the adjuvants, MPLA, CpG, all used widely in US approved vaccines and pharmaceuticals. And second, we are teaching people to do what successful people do by themselves. We are teaching people to attack the same targets as SARS-1 survivors attacked with their T cells. And so we see those people thriving. So we don't believe that T cell attack against those targets is dangerous because people have been doing it for 17 years. There's literature on that. And so we're simply teaching you to do what you should be doing on your own. And that's almost a very organic way to look at this. We're not reprogramming anything. We're not changing DNA. We're not changing RNA. We're just exposing you to a peptide, making your body think it's important by adding these little adjuvants that cause the immune system to wake up, and then letting you naturally target killer T cells against that. What is that phenomenon? It's the same as successful folks who do that naturally. What about booster shots? You would hope that if we pick stable targets that aren't changing on nucleocapsid, you'll never need a booster shot. Why? We know that T cells last for 17 years after SARS-1, so it lasts a long time. And as long as you're hitting things that aren't changing, maybe you never need a booster. And that's our thinking on this. Proofs in the pudding, that's our thinking. See, I've linked to the video in the beginning. We have a similar video on the website. I'm going to ask our technicians to put the exact same video you just saw on our website. So that's on www.strategicvaccines.com. And we'll put the same up. We'll put the exact same video up there. The one up there is similar. We'll put the same one up there later today. what vaccine have I been inoculated with? I got the Mrna simply because it was the first one I could get. I got it from one of the hospitals I work at. It was available in December last year, and I took it as the first one available. My order of preference would be Pfizer first, Moderna second, J&J third. I think Pfizer is marginally better than Mrna in terms of the immune response, but I don't think it's that significant a difference. I think the 2 mRNA vaccines are probably significantly better than J&J. The other problem with the J&J is because they're using an adenovirus vector to give the DNA that programs your cell to make the spike protein, because they're using an adenovirus vector, it's hard to give a boost because your body will tend to reject that vector the second time you get it. So the J&J vaccine is kind of not designed for a boost. So they're kind of stuck. The mRNA vaccine's easy to boost with. You simply load a new RNA fragment, generate a new version of the protein, get an antibody response. That would be great, except that, of course, we're concerned that antibodies are not the answer here long-term because of the fact that spike can mutate so effectively, it may be able to completely eliminate the response. What are the side effects of your T cell solution? We hope none. Why is that? Healthy people that made a response to the virus on their own produce T cells with the same targeting that we're using. Every component we use on the platform is already used in vaccines. We hope there are no side effects. The goal of phase 1, human healthy volunteers, give them the material, look for an immune response, is to answer that question definitively, but we hope there are no side effects. What remedies, if any, are available to reverse long-term effects of past COVID, like loss of taste and smell? Very interesting question. Our approach here is to kill the virus, reduce viral load, and prevent a lot of this stuff from happening. We hope that if we come to somebody who's freshly infected, we wipe the virus out, they won't have any long-term effects. Can it reverse loss of taste and smell? I don't know. But I think the best thing you can do if you're infected is get rid of the virus. Now I'm going to throw in a medical pearl that I believe in, which is not commonly done. It is my belief, going on record here, that if you get COVID, you should be on anticoagulants for at least a couple of months. We are seeing these horrible blood clots occur quite a few weeks after people recover from COVID. And I think that you should be on one of these new, perhaps oral anticoagulants like Reid Rubsamen, which is Xarelto, something like that, because the last thing you want is a blood clot. Our medical community is reluctant to do that because these things have risk. I'm telling you, if you have a knee replacement at any hospital in the US, you're on that drug anyway for a while. Why not give it to COVID patients? It's controversial. I'm sure people will disagree, but that's what, if I had the COVID virus, I'd be popping those pills for at least a couple of months because stroke is terrifying and it's a significant risk here. Okay, I'm just reading on here. How can you be sure the T cells are targeting the virus only and it won't cause the immune system to go into overdrive, attack healthy cells? We addressed that. Very good question, because we're mimicking what people do naturally. At least some people do naturally. When they attack these targets, they thrive. They don't seem to have any side effects from naturally targeting these same targets. So we feel this will be safe. It's a very good question. What evidence is there about natural immunity? People have prior infection and now have antibodies work or doesn't work. Why is there so little discussion of natural immunity? I can tell you from personal experience with patients, natural immunity is not perfect. We all know patients that have gotten the disease more than once if they were not vaccinated. The vaccine does produce a better immune response generally than natural immunity. So, you know, is natural immunity good? Probably provides some protection, but you need the vaccine. You should get the vaccine. You should not rely on natural immunity. What is your opinion of PCR test? Is this designed for COVID testing? PCR is interesting. Polymerase chain reaction. So how does it work? PCR is designed to amplify small quantities of DNA. So you have to do a bit of a trick. COVID is an RNA virus. So you have to convert the RNA to DNA. There's a way to do that. And then amplify that to see it. It is a very sensitive test. I don't know how much detail I should go into here, but there is a key metric, which is typically not reported, which is called the cycle time. So if you run a PCR test, the cycle time is how many times did the machine have to cycle to find the signal? And most people agree, well, if it's like below 30 times, you probably have real virus. If it's a whole lot more than that, you probably don't. But the difference is soft. It's not really agreed upon. And that's important because these things are super sensitive. And the other problem you have here is that you may have dead virus in your body for a while. and you may get a positive PCR test even after there's no live virus. So we don't have a good test for live virus. We don't have a cheap, inexpensive, fast test that says, do you have live virus? We only know if you have nucleocapsid antigen, which is this rapid test, like a pregnancy test that you stick in your nose and you look for the bars on the cardboard. That's an antigen test looking for nucleocapsid. We have that. And we have PCR, which looks for, again, the RNA, which is converted to DNA and then amplified. But you can have that RNA in your body and not have any live virus. So that's imperfect. You'd rather know about live virus. So you might have a positive PCR test and not be contagious, but it's the best we have, and it's certainly very sensitive. So in science, we say it's a sensitive and not specific test in terms of live virus, but it's the best we have. What's holding up the prosecuting a warp speed effort for cellular T-cell vaccine? What's holding us up is no human data. We cannot get this cornucopia of government money, which is potentially available for scale up here and warp speed type development, without presenting to the government human beings who've received the drug. And the first course of action there is to take healthy volunteers, give them the material, the drug, by inhalation, take blood samples and so on, and see if we got an immune response, which we expect to get. We got it in primates, giving it the same way. And then look for side effects, which are very important to look for. I mentioned before, safety is number 1, 2, and 3 on our list. And then report that data, publish it. With that data, we can at least apply for extensive government funding. And we created the public-private partnership at Strategic Vaccines to enable that, because that kind of mechanism is important for accessing funding from these government sources. How early do patients have to take your drug when you get COVID to be efficient? Is it sufficient to have the drug after you already have symptoms? Really great questions. The earlier, the better, okay? This is a COVID, what does it do? It produces cytokine storm in the worst case, which are cytokines are very toxic chemicals the body releases to actually enhance the immune response, but they can be quite deadly. So the earlier, the better. One of the reasons I think you saw bad results from some of the therapies that were tested earlier during COVID is they were given late. The FDA likes to say, well, we're not sure if it's safe, so give it to really desperately ill people. That's a problem. Antivirals in particular work best early. If you give them late when the cat's out of the bag and all these toxic chemicals are released, they can be useless. So you really need to give it as early as you can. I don't know how early that would be, but again, earlier the better. Can you give it after you have symptoms? Yes, you could, and we want to do that. But again, when you already have symptoms, it's gotten later in the day. So the wide availability of testing has really made it possible to even think about giving a material like ours early. And we hope that that will be the order of the day here as we go forward. What about the number of deaths reported in the CDC's various systems taking the vaccine? Understand excess of 10,000. Most vaccines have been stopped by now, and the number of adverse events, vaccines understand number excess 250,000 reports. Why is this not being discussed? It's on the CDC VERIS system. You talk about adverse events producing something really bad and long term. The vaccines have been very safe. The reports of blood clots in the J&J vaccine occurring in a few parts per million. Well, that of course is concerning, because you worry, hey, if they're catastrophic in persons per million, could they be less catastrophic and more? But even that has not really panned out to be a big concern. The FDA put a black box warning on, or a warning on the package insert, but that drug has not been discontinued. I don't think it's true that there have been significant serious side effects from the mRNA vaccines. When one of my professors, I was studying at the public health school, I'm very interested in public health and the effects that all our approaches we have to be synchronized with public policy and public health. And one of the guys there is the attorney for CVS, and he told us after 800,000 doses, the first 800,000 doses of Pfizer, they had 0 adverse events there. I think these are very safe. I don't think we have to worry about the mRNA vaccines. And I certainly think that everyone on this call should take it if you haven't already. My whole family has had it. My 15-year-old son got it. My 21-year-old son, who's a firefighter EMT, who got it actually very early. My daughter is a nursing student. She got it. My wife and I got it. So I think she got Pfizer, I think, and I got Mrna, and other kids were split too, all based on what was available at the time. So I don't think it's a concern. Again, look, I mean, I'm not belittling side effects at all. I'm just saying that the risk here is so ginormous, and the effects we've seen have not been extreme, except in very rare cases. And of course, you always wonder what was the cause of that. So I really think these are safe.
Reid,
Clarify.
wait, Reid.
Yeah, sure.
As an advocate for your son, I'd like to interrupt. He's
Yeah.
also he's also on his way to medical school.
That's true. He's majoring in chemistry and
Thank you.
philosophy, and he wants to go to medical school. And he's a great kid. All my kids are great kids. I don't know where they came from or what role I had, but my wife says she raised them all as in the lab. She was sure she could say that right now. Okay, to clarify, Pfizer, Moderna, J&J vaccines. I don't quite understand the question, but yes, we're talking about Pfizer mRNA vaccine, Moderna mRNA vaccine, J&J. What did Pfizer do? They put in RNA that programs manufactured by your body of all the spike protein. Moderna said, we're just going to do the receptor binding domain because that's what binds to ACE2. Two different approaches. The immune response from Pfizer is probably a little better than Moderna, but not hugely so. J&J, probably not as effective as the other vaccines, and has this interesting property that it's hard to boost with. Or they should be, at least in general, adenovirus vector vaccines are hard to boost with because your body makes antibodies to the viral envelope, which is not COVID, but this inert virus that's used to inject the DNA to produce the protein. And that makes it hard to give a second dose because your body will tend to wipe out the carrier before it has a chance to give you a second dose. How can an investor invest in your company project? Please contact us on the website and Henry will get in touch with you. What do you need from us? Again, please reach out to Henry. Will this product be financially available to everyone? I'll let Henry answer that question. Why is strategic vaccines the only firm talking about relevant T-cell vaccines? Does it take longer to produce such a vaccine? Really good question. We're not the only company talking about T-cell vaccines. There are others. We are the only one in advanced development. So we're the only ones that I know of. Educate me if I'm wrong. I've been wrong before. Ask my wife about that again, but ask my kids about that again. But as far as we know, we're the only people with primate data showing that our T-cell vaccine works to suppress symptoms in primates exposed to high doses of SARS-2. That's all I know. But does it take longer to produce? Actually, it should be shorter to produce. So our vaccine is made synthetically. It doesn't require, a big soup of virus like J&J has to do for their vaccine. You may have seen that they were using emergent biosolutions to make their vaccine and got cross-contaminated with a different vat of something else. And when you have these live things, brewing, it's kind of like a witch's brew in a way. So it's not nice to say, but it's just fundamentally, you're talking about a situation where it's hard to manage these big processes, expensive, complex. You've got living systems doing stuff. RNA is hard to make because the raw materials are hard to come by. We make these materials out of peptides and polymer, easy to source. The adjuvants are straightforward to source as well. So we're actually using components that are easier to make, faster to make. And we made this product ourselves in our lab and now at Oakwood Labs here in Solon very easily. So it is a fast, and also I should point out, much faster to make the antibody therapy, which is also difficult and complex to make. Side effects to date, what are the side effects with cytokine release? So we looked at this very, very carefully in animal models. Our published article in the journal Vaccines, which is a Swiss journal, again, an index journal, well-known journal in the vaccine space. We looked at cytokine release after giving the T-cell vaccine to primates. We did not see, for example, differential. So they're Th1 and Th2. This gets kind of complicated, but there's a Th1 and Th2 T-cell response to this kind of thing. And what you'd like to see is you don't want to see an asymmetric increase in Th2 response, which could produce inflammatory side effects. We did not see that. We did not see any toxic cytokine release associated with vaccination. For example, we did not see release of IL-6 associated with the vaccine versus control. So we looked at this very carefully. It's a very good question, by the way, very good question. And it really is one of the core safety questions you have to ask with a T-cell vaccine. I would guess this person is an immunologist who asked this question, Scott Akers. But anyway, it's a good question, Scott. And we looked at that carefully. Please read our article. Senior author is Paul Harris. I'm the corresponding author. It's in Vaccines, VACCINES. You can look it up on PubMed and read it yourself, but it's all up there. Peter Workman, thanks for answering my questions. Thank you for clarifying your peptide is peptide-based. I should have done that earlier, Peter. I apologize. Mitch, do you know Immune Bio? Any thoughts of your products, target information? Scott, do you know Immune Bio?
I do not.
Okay, I don't either. So we can get back to you on that. We don't have that on our radar. Would you kindly provide for recorded playback of this? Thank you for calling an educational session. Thank you. Sure, I know Michael is recording it, and I'm sure he can provide that for you. Please reach out to Michael Corcelli. We had a big 1918 big pandemic, almost 100 years, COVID 2020. Of course, this person is talking about the influenza pandemic that most people don't know killed more people than World War I did in casualties. So there was a huge pandemic with a very serious aftermath. Do you have some sort of artificial AI system to project the next big pandemic based on data and what you're coming to mitigate? This is a very good question. So thank you, Zalim, for asking it, Zalim. We wrote an article, Scott Burkholz was the senior author, I'm the corresponding author, about the movement of this virus from animals to man, in particular minks to man. And Scott and I and our co-authors at Stanford believe that one way we really need to be thinking about future pandemics is to look at animal reservoirs of the virus. The virus is able to jump from humans to people and back and forth, and humans to animals and back and forth in a way that increases the efficiency of its ability to mutate and create more variants. So this is a big issue, and it's a big opportunity, because if you went ahead and sequenced agricultural populations, pets, and so on, you might learn a lot more about this. Now, there's a funny thing happening right now. A company that we're familiar with personally developed a vaccine for COVID for animals, for pets, and USDA wouldn't authorize it. And apparently it's because they didn't want to scare people to think their pets could have COVID. And the United States is not aggressively sampling agricultural populations of animals to look for viruses. In fact, the mink study, there's only a few farms in the U.S. that provided data. A lot of it came from Scandinavia, actually from Denmark. So you know, we don't seem to be in a hurry as a country or as a planet to look at animal populations for gene sequencing the virus, but that actually may be the best way to look for the next big pandemic. And that's just an opinion that we have. But the way we use AI today is to predict how a particular virus is going to behave. So we did quite a bit of work on looking at the, not to use too many buzzwords here, but the thermodynamic stability of proteins within virus. We did a lot of this work with Zika virus, for example. We repeated this with Ebola and also with COVID. We published our results on our Zika virus analysis in Frontiers in Immunology. You can read that. Dicio Cunaneto is a senior author. I'm the corresponding author. That was published in 2016. It's on PubMed. But that article describes how we use computer modeling to predict, in a simplified way, what are the crowded parts of the virus protein that really can't change. If you're packed in, making one amino acid change for a different one can be hard. So if you can predict what those sites are, and you can target those, you're more likely to have a longer lasting effect of your vaccine or therapy. So we use AI to do that. We also use AI to do another important task I alluded to before, which is how do you make a T-cell vaccine? Antibody vaccine doesn't care who you are. It's just there to bind the virus. We have to care about who you are. People from different countries, Africa, Latin America, United States, Asia, they have different HLA types. They have different immune system characteristics. We have to make sure that any individual across the world can see our vaccine and recognize the peptides. So we picked peptides, all these, only 3 actually in this model we're doing now, that are predicted to have very, very wide worldwide population coverage based on AI modeling, but also for belt and suspenders, we did not want to rely only on computer modeling. So we picked 3 peptides for which there is extensive in vitro laboratory data showing the HLA binding affinity for each one of those peptides in human cells. And this is important because we now can calculate, we think very precisely, that we can cover essentially all the world's population with this design that we made. And we think that we can cover everybody with it, and we'll find out obviously going forward. But that's an important consideration, and AI turns out to be useful for the thermodynamic part of this, and also for finding these favorable targets and predicting which ones are going to be the best to attack based on their lack of ability to mutate. Last question, Peter Workman, thoughts on hydroxy? You know, I thought hydroxychloroquine was a lot of us thought it was interesting because in vitro, it's known to affect spike binding to ACE2. That was described pretty early on. Well, gosh, if it did that, it could be effective. There were a couple of controlled studies done looking at this. I never, one was done at WashU St. Louis. I forget what the other one was done. It was done with the high-risk healthcare workers. I don't know if those results were ever published in final form, but I don't believe any signal came out of those showing benefit. There was a lot of talk about hydroxy being deadly. It's actually pretty safe if it's given a low dose. There's also discussions about patients on Plaquenil who have lupus. So Plaquenil is a very widely used drug for lupus, and basically talking about the same thing, the hydroxy type of thing. And basically, there was initial data that people with lupus had less COVID. I think that's pretty much been dispelled. So I think that was an interesting question because of the in vitro data that's showing there was this inhibition. But I don't think we ever really came away realizing this would be really great. Remdesivir, of course, was approved as an antiviral agent very early on in COVID. It was developed for MERS, Middle East Respiratory Syndrome, by Gilead Corporation. It's intravenously administered, and that was marginally effective. Again, it was given probably too late. The most interesting, I think, therapeutics early on were the neutralizing antibodies in the bag. So you can make monoclonal antibodies and put them in a IV bag that bind spike. And so some folks at Regeneron and Lilly made these things, and they were pretty interesting initially. But of course, what's happened is these variants aren't as susceptible to them. And one of the products was recently approval was withdrawn by FDA because they found it to be ineffective. So these things that started out working pretty well are now weaker because of the variants. And again, the monoclonal antibody is just a precursor of what vaccines are going to do. Vaccines produce an antibody response. You don't know which antibodies someone's going to make to the vaccine, but they'll make a lot of them. But the extent to which the virus is mutating, those antibodies will become less and less effective over time. And of course, we already said that antibodies given in a vaccine only last a short time. It's also interesting, just my closing thought, it's interesting that of all the bags of antibody that were made, and there were a lot of them made, the government paid a lot of money for this. I think it was on the order of $1 billion, I forget, but very few doses were ever given. I think it was a vast minority were given. Well, why is that? This is funny. I go to the hospital 8 days a week. I do pay attention to things when I'm there. It's funny when you practice even a little medicine as I do, some things are just obvious. Like it is obvious that we don't have a mechanism in the United States for giving an outpatient an antibody infusion. I mean, we have infusion centers for cancer chemotherapy. They're expensive. They're not many beds. It's an all-day affair. You can't just have somebody walk in your clinic, they're positive, and give them an antibody. It's not easily, you can't admit them to the hospital to do it easily. It's not cost effective. There are not many beds. So it becomes a really non-viable therapy. You need a way to have a non-refrigerated, non-intravenously required therapy that works. And when you're talking about antibodies and remdesivir and other stuff, it just gets too hard to manage in the real world with patient after patient after patient coming to somebody's clinic and wanting something today. And the answer right now is, well, go home and get a fever, call us back. Well, that's not what people want to hear. They want a therapy. They want a treatment. They want to go home with something. And I think that we really focused on this inhalational delivery because the lung is the first line of defense for respiratory disease. Also, the Army was very adamant that they wanted their Marburg vaccine given by inhalation. They said, look, we've done a lot of research on this. A lot of it was never published because it's classified. But they said, look, you need to, if you want to protect people from these deadly viruses, they're transmitted by air, you've got to protect the lung because that's the first line of defense. You don't want it to get through the lung and then manage it somewhere else in the body. So they were adamant about this. And I don't know if I mentioned this earlier, but they called me up early COVID, said, Reid, get off your butt and go make a COVID vaccine using the same technology. So, would you have any money? They said, well, no, it's all in warp speed, but do it anyway. So we marshaled our resources and I took my begging bowl over to Oakwood Labs and said, I don't have any money, but we'll give you some stock and we'll pay for the chemicals. Can you make this for us? And they did. And they worked very hard for a year and they put 20 people on it. And they worked, 7 days a week really and did it for us. And I'm very grateful to them. But really, the Army influenced our thinking quite a bit on this because of their work and their real, belief that you need to give these important vaccines by inhalation. So that's where we are today. I'm going to look at there was also a chat room, a chat thing, and I want to see if I think we covered all the ones in the Q&A section. General use of funds breakout. Robert Corleone. Okay, fine. Is that, do you see that, Michael? Should we?
I'm actually looking.
I don't know what that means, but we're more than
That-
whatever that is, we'll deal with it.
That may have just been sent to you. I don't.
Okay, it says general use of funds breakout question mark Rob Corleone, and there's an email address which I will send to you. Okay, so I don't see anything else. Oh, there's a master chat. Let me see what's on the chat. Is there evidence to suggest that SARS-2 has a latent phase as HIV? Okay, very good question. And yes, and in fact, the long haul syndrome may be a quasi-latent phase. Of course, obviously you're feeling sick, but it may be a viral reservoir, which is the latent phase of HIV. Very challenging, but it may actually be susceptible to those killer T cell attack. Doctors, thanks for doing the webinar. You're welcome. What is the timeline for other treatments, T cell approach, available? How scalable are they? Very scalable. We are working, I mentioned Lockheed Martin, the Warp Speed guys in College Station, Texas. We've already purchased a significant amount of equipment to do this at a very high scale. Labs is ready to make more right here in Solon, Ohio. What is the timeline? We have drug ready to go right now for human testing, 800 vials sitting there in Solon across from the street from my house, half mile down the road. We're going to use that in phase 1 in Australia. We're going to make more for phase 2. We're only talking about 300 subjects for phase 2, so not a whole lot more material is required. When is it available? Gosh, if you look at what happened in the first part of the COVID epidemic, it's all up to the government, but it could be available very quickly. We could have this fielded and ready to go in very, very big quantities early next year. But a lot of that's a regulatory factor, will depend on the state of emergency and the status of the disease itself. Will antibody vaccines, which is vaccines, work together? Can we use this with an antibody vaccine? Absolutely. So one thing I'd like to look at here is what happens if we boost people who got an antibody shot with our material, okay, and create a tandem T cell response. Could be very important. This could be in fact, if you want, maybe this is too bold here. Henry tells us to be more bold. I'm not bold enough, but look, I mean, we could be the savior to the antibody vaccine industry. I mean, they're sitting there trying to pump these boosters out, playing whack-a-mole with a new booster every, I don't know how many months. They haven't gotten the first one out yet. We could theoretically dose all those folks and protect them against the variants. So there's nothing about having a previous mRNA vaccine to prevent us from getting involved. We already know mRNA vaccines produce a T cell response to spike, which is good. But with our FLOVID-20 in there, maybe we could produce a T cell attack against nucleocapsids. So I see this as potentially very synergistic.
Reid, could I jump in for 1 second?
Go.
The machines that have been optimized by Lockheed,
Yeah,
can be set in airtight containers all over the world in a C2 if that's the right number of the plane, and they produce about 1 million doses a month.
well, we're fortunate to be working with an aerospace contractor, and they actually sat down with us and actually measured the sterile modules that we use to put these machines in, and they actually can be moved easily around the world. We actually got some inquiries from countries in the Middle East in particular interested in possibly participating in manufacturing this material later on. And I think it's very encouraging that, again, this is a simple process. This isn't like, these big vats of stuff you have to have for recombinant protein manufacturing like Novavax has to do, or antibody therapies like Regeneron and Lilly are doing, or adenovirus vaccines like J&J is doing, mRNA vaccines with complex minus 90-degree cold chain requirements like Pfizer has to do. It's simple to make, simple to distribute, simple to store. So we think we're in good shape with that. Thank you for the presentation. Can you comment on side effects, risks of current mRNA vaccines populations that aren't at high risk? What are your views, potential long-term risks? Some have highlighted blood clots, autoimmune spike protein shedding, et cetera. I tell you, I believe all marketed vaccines are safe. I think take any one of them. I think the mRNA vaccines are probably better than the J&J for a bunch of reasons, including better initial immune response and the ability to boost. But I think you got to get it now. You do not want to be walking down the street unprotected today with this Delta thing out there. This is a deadly virus, 1,000 times higher viral load than the original one. If you ignore this whole video and forget the whole thing, please get an mRNA vaccine today. Please do that. I'm not benefiting from that in any way except knowing that you're going to be better. Will we have access to this recording link? This amazing talk. Thank you for saying that. Yes, Shelley would be happy to provide that. Okay, this is the same question I already answered on QA about AI and the 1918 pandemic. What is the best vaccine to get right now? Moderna, Pfizer, J&J. As I mentioned before, my mental order is Pfizer best, Moderna second, very close second, essentially the same. J&J third, all safe. But I think the J&J one a little problematic because it's probably going to be tough to boost it. The initial efficacy isn't as good as the reported efficacy of Pfizer and Mrna. I think I took Mrna. It was more important to me to get it faster than to get my first choice because I don't think there's that much difference between them. But my whole family some got Pfizer, some got Mrna, all based on what was available.
Reid, can I add one more thing?
Talk to me.
I've had the Pfizer, and everybody in my family, of course, got vaccinated as soon as they could.
Yeah.
It's still really important to wear a mask in public.
Look, I was out and about today. I was buying a takeout breakfast before this talk, and I wear a mask in public. Look, I mean, I think that we're really, unfortunately, entering another big phase of worry here. Like I said before, if half the people are vaccinated are getting Delta, you just don't want to get it. Well, gee, you won't get that sick. Well, maybe you will get sick later. Maybe you will get brain fog, cardiac inflammation, blood clots. I don't know. Nobody knows because the Delta hasn't been around long enough to see what the 90 days, 6 month data collection is really going to be. So I think you got to be very conservative. I think, and of course, you can be vaccinated and shed virus and infect your friends. So many of my friends are not vaccinated.
There's one more thing that's okay to point out when somebody asked about the CDC. There really is, and it's pretty obvious. There's a mismatch between what we're told and what some people might know or what some people don't know, and it's very confusing the way all this misinformation has been disseminated in public, and misleading. And so that is another problem, and it does create anxiety, a hell of a lot of vaccine angst, and confusion is never a good idea. So,
let me get to one more question here, Henry. It's almost done. Some epidemiologists are worried that the current vaccines could cause ADE long term. What are your thoughts? Very good question. Thank you for asking it. I was not going to cover this, but it's worth spending a few minutes on this. What does ADE stand for? It stands for antibody disease enhancement. ADE is the reason we do not have a vaccine for respiratory syncytial virus, and we don't have a vaccine for dengue fever. Because what ADE, and I'm going to simplify this, but ADE basically is, okay, if you have an antibody against the virus, it actually makes the virus worse. It kind of drags it better into contact with the cell, for lack of a better explanation. So an antibody that is not neutralizing is at risk for ADE. One of the reasons the guys who make antibody vaccine did not try to make an antibody against nucleocapsid is there is a theory that you could get ADE if you had an antibody directed against nucleocapsid. And so that even though I mentioned the antibodies can't really hit the middle of the virus, nobody even really tried because of that risk. Now, our vaccine does not produce an antibody response. A 9-amino acid peptide will only produce a T cell response. So that's not a risk for us. So we were able to play in this sandbox where other people could not. So would the current vaccines cause ADE? I mean, this was a concern, okay? Because so you have a neutralizing, you have a vaccine, you take it, antibodies are neutralizing. The virus mutates, they're not neutralizing anymore. Could they cause ADE? Theoretically, yes. We don't seem to be seeing it. A lot of smart people are following this. This is a well-known risk. I read about it. Our group follows it in literature. Folks looking at this in various countries that have problems are not seeing evidence at ADE. So I think we might have dodged this bullet and not had this problem, but it's a good question and it's a concern. I mean, I certainly got the vaccine. My family did. I mean, I know this is a risk, but I think the protection outweighs the risk. But you're right. It's a theoretical risk if what was once neutralizing isn't anymore. This is another great thing about T-cell vaccines. There is no ADE risk. And actually, when I pitched BARDA, the first part of the epidemic, big call, hour talk. Everybody was there, FDA, BARDA, military, all the people with money, people with knowledge. It's amazing. I was on a Zoom call. And I give the pitch and I said this. The funny part about that pitch was, I'm like, well, what do you think? And they're like, Reid, spike is not going to mutate. Antibodies neutralizing. We got it covered. Have a nice day. And I'm like, no, I mean, it is going to mutate. There's a lot of literature showing how it probably will. You need a plan B. No, we're good. So I mean, you know, at some point you don't know what to say, right? On the other hand, we're like, okay, fine. We didn't have the animal data at that point. We'll get the animal data, we'll publish the articles. And I really want to emphasize publishing the articles. We developed our T-cell vaccine platform in 2014, peer-reviewed journal article. We did analysis using AI and bioinformatics to find weak spots on viruses, peer-reviewed journal article. We ran a test to see if it would work against Ebola, peer-reviewed journal article. We did an analysis of our COVID vaccine in primates, peer-reviewed journal article. We looked at, what's happening with people and animal transmission? Does this explain how we get more efficient mutation? Peer-reviewed journal article. Could spike continue to mutate enormously? Could it become a big threat? Peer-reviewed journal article. We also wrote an article parenthetically about sort of after the fact therapy. How do you have antibodies against these horrible cytokines? We did an extensive, one of the earliest, actually the only experiment I think ever published on attacking IL-6 receptor, IL-6 antibody and stirring cytokines formed from Ebola. And some interesting results there as well. But of course, that's late in the phase. As we said before, that's after the horses out of the barn. But we've published extensively. It is interesting that when Mrna went out with their phase 1 study, they had 0 peer-reviewed journal articles in the history of the company, not a single one. And they've been in business for years doing cancer vaccines and so on. So we have a very different approach. I think, look, I'm a physician scientist. I'm on the faculty of what I think is a great medical school here in Cleveland. And I'm trying to learn, trying to teach, and trying to learn myself, educate. And if you're not publishing, you don't get the yeasty, you don't get the commentary, you don't get the transparency, you don't move forward. You don't really feel like you're contributing or learning. So I think it's very important to publish as you go. It's of course, it's time consuming, and articles come out later than the work as you're trying to go to the next thing. And we work very hard to publish these things, and we still have papers in the pipeline, but it's not trivial to do. I think it's very important. So we do that. Last question on the list. Do you know thin film? Of course, I won't be able to answer this one, but I'll read it anyway. Do you know thin film freezing pharma, TFFP, in their lung delivery system? I do not. Scott, have you heard of TFFP, thin film freezing pharma?
I have not.
We will look that up too. It's the same question we couldn't answer today. I guess that's low percentage
like
ones we couldn't answer.
Cryogenics, freezing human beings, and they'll come back in 50 years.
No, I don't think that's what it is. But anyway, so there we are. So I'll turn it back over to you, Michael. And thank you for, let me go back to Q&A, see if anything else came up there. Oh, there's a bunch more Q&A questions. Again, the freezing pharma again, we'll check that out. Benefits of ivermectin. Interesting question. Don't know a whole lot about ivermectin. There's some interesting folks have looked at that. Of course, it's one of these controversial things. I just, Scott, is there any new signal on that we should know about? I'm not aware of any, but
I've just heard there's possibly some efficacy and it's being discussed.
yeah, I mean, again, we're scientists. I mean, I don't care what's controversial, what isn't. I mean, we're looking at the data. Again, publications are important, and I'm not aware of anything big and new about it, but we're definitely, a lot of people think it's great. And, it's funny, people argue against anecdotal reports. Every important medical study ever done in the history of man started out with somebody seeing something on a one-shot out, right? They're like, well, I tried this, it worked. Then they do some big study. So I listen to people when they say, hey, we tried it. I mean, I think it's important to listen and not get freaked out by whoever says it's terrible or great or whatever. So we listen to everything and we read everything. So I'm not aware of anything big in that area. New last question from Mitch. CDC Director Walensky said recent studies show those vaccinated individuals who do become infected with COVID are just as much viral load as unvaccinated. How should we think about vaccines efficacy? It has long been known, hypothesized and known that the vaccines may not do a whole lot for viral load. In particular, you may be infectious even if you yourself have no symptoms. And this is recognized pretty early in the whole, SARS vaccine effort. And so there is this problem of, you could still infect somebody even if you're not infected. Now we have our COVID study in primates was not a large study. It was eight animals, 4 active, 4 controls. It's not the biggest study history of man. If we use all the primates like that, this is interesting. There are no more primates in North America. No more rhesus macaque primates because they were all used by the COVID-19 pop-up industry that occurred because they're critically important. So most of us in the space are not using cynomacaques, which are not exactly the same, but it's all we have. So our study wasn't big. We didn't have access to more animals. But so we did show in the majority of the animals that we did back down. We did do a nice job of suppressing viral load with this lung-delivered vaccine. In fact, we suppressed it in the nose, which kind of, it's kind of my belief is that the respiratory tract, it's kind of the nose and the lung are kind of interconnected, and it appears like it's one sort of lymphatic immunological ecosystem. So it kind of makes some sense to me that if you get an immune response in the lung, it affects the nose and vice versa. So it may be possible to give our drug nasally also, that we're not testing that currently. We're testing that with other studies we're doing, but not with COVID. So fundamentally, the vaccine doesn't guarantee, the antibody vaccines don't guarantee you're not shedding. We hope our vaccine will suppress shedding. We don't know yet. We're going to look at that in our studies. But it's a good question, and it's something to keep in mind. So that's, I think let me go back to the regular chat room and see if I got any more questions during the talk. Oh, thanks so much for the questions. Here's another one. Is there anything folks who receive J&J do? Look, nothing wrong with the J&J vaccine in terms of a safety standpoint. You probably can't boost with it. You could certainly get an mRNA vaccine behind it. Is there a risk to that? Well, you always wonder, is it, if you're quote unquote boosting the J&J with an mRNA, will you see a bigger response? I think people have already looked at this. I'm pretty sure there's been hybrid reported studies of J&J and mRNA. I'm not sure, but you can certainly take an mRNA vaccine, I believe. Although don't take me here. What do the talk show doctors say? This is not medical advice. It's just an idea. So, but I'd have to look at that. I can look at that and get back to you. Loretta, I can look at that today and get back to you. And I think that there are some countries looked at splitting J&J and mRNA, and I can check that quickly, but there's nothing, I wouldn't, lose sleep over it. I think, the mRNA are probably better, and one could consider getting that, as I say on the medical talk shows, talk to your doctor, but I'll be happy to read about that. And I don't know if I know how to get back to you. I'll ask Michael Corcelli.
We'll get that to you.
Okay, we'll figure that out. That is all we see. All I see here.
See the one.
Wait. There's a new one. There's a new question.
COVID.
New question. Wow, we keep getting more questions. Contact. Okay. If you had COVID, would you still recommend the vaccine? Absolutely. You need the vaccine if you've had COVID. We know for a fact that natural immunity is not as good as vaccine immunity. I strongly recommend you run out if you've had COVID and get the mRNA vaccines. Mitch, statistics show countries with the highest COVID per capita, the same countries, highest population vaccinated. Their rapid declines. I'm trying to understand the question. Statistics show highest COVID, highest with vaccinations. Just a few months ago, these same places were explaining the rapid declines of the vaccine. I'm not sure I totally understand the question, but I think it's saying something like, if there's lots of vaccinations, it's the highest hospitalization per capita. I'm not sure if those statistics slice and dice quite like that. Israel, of course, has had an incredibly aggressive vaccination program. And they did that because they have an incredibly high incidence rate of the disease, largely because they have a very concentrated, population living situation. And they reported 50% of the vaccinated people are the ones with the 50% of the new infections are vaccinated people. So that may partially answer that question. But fundamentally, I don't think that the vaccines are dangerous. I don't think the vaccines increase your risk of getting an infection. I don't think they produce ADE. A lot of smart people know what ADE is and are following that data. We follow it, whether we're smart or not, we follow it. And we're not seeing any published evidence of ADE. So I really can't think of a good reason to not get the vaccines. And whichever one you got, I think, it's great that you have it. And I think there's no question that if you're naked without a vaccine right now, the Delta is really dangerous. And again, 1,000, you don't have to think about this too hard. 1,000x viral load with Delta versus the original. Not a lot of reason to not be really worried about it, right? And again, I use the battering ram analogy. These variants are just banging away at the door, and eventually they will crack through the vaccine door. And when they do, it's going to be a bad day. And it's going to be a bad day because we're going to see rising deaths, probably in some peripheral country that originally wasn't well vaccinated and is exposed. And the worry is that we're going to see some real issues here, unfortunately, fairly quickly. And I hope we have some more breathing room. We'll do the best we can, but fundamentally, we have to think about other solutions than just antibody vaccines here. Back to you, Michael.
All right, so that concludes our discussion today. We will be following up with you within 24 hours with a recording of today's fireside chat. We hope that you enjoyed it. I strongly encourage you to reach out to the company if you have any questions or like more information on the alternative investment opportunity of the century. So please strongly consider it. Have a heart, and also participate in the financial success that Strategic Vaccines is playing out right now. So guys, thank you very much for being a part today. Thank you for all of you who came and stayed till the very end. We appreciate your time and attention and consideration. Have a great day. That concludes
Thank
today's
you.
Fireside
Thank you,
Chat.
Michael, for setting it up. And thank you, Henry, for being here. And Scott and Kevin, thanks so much.
All right. Thank you,
Thanks,
guys.
everybody.
Have a great day.